Hi, my name is Kari Soltau, and I am currently a member of CORE. CORE stands for Collaboration on Riverine Ecology. The collaboration is between mathematics and biology. Within this program, I myself, a biology student, was paired up with another student of mathematics. We were then asked to investigate an important host-parasite interaction occurring along the Mississippi River. This interaction has caused detrimental effects both economically and ecologically, with over 60,000 waterfowl deaths since 2002. I would like to share with you a little background information, an overview of the life cycle, and the establishment of Team DEB. This is a photo of Bithynia tentaculata, an invasive aquatic snail that was recently introduced into pools 7 and 8 of the upper Mississippi River. Along with its shell, this snail harbors two trematode parasites, Cytocathaly buccinus and Ceridiotrema pseudoglobulus. These parasites have a complex life cycle that utilize 3 hosts, each of which harbor a different stage in the life cycle. Alright, now I would like to go through the life cycle of this parasite. So things start off when the waterfowl consumes Metasicaria. When these Metasicaria have reached the intestines of the waterfowl, they then develop into an adult trematode. After developing into an adult trematode, these worms produce eggs, lots of eggs, which are then passed from the waterfowl via their feces into the aquatic environment. These eggs then develop into free-swimming Miracidia, and these Miracidia are ingested by the first intermediate host, which is Bithynia tentaculata. Once they're in Bithynia tentaculata, they develop a little further, and what is known as cercaria are shed from the snail. Now these cercaria are now looking for the second intermediate host in the life cycle, which will be either Bithynia tentaculata or another native snail species. Once they've reached the second intermediate host and penetrated it, they migrate up to the spire of the shell, where they then encyst and form what is known as the Metasicaria. After this point, the snail may be consumed by a waterfowl, and this would continue the life cycle. Now I would like to talk to you about Team DEB. Team DEB consisted of one biology student, myself, and one mathematics student, Casey. Together, we decided that we wanted to observe energy allocation patterns in Bithynia tentaculata. In order to do this, we first set up an empirical study in which we observed infected and uninfected individual snails over the course of 10 weeks. Each week, length, mass, and volume was taken for every individual within our study. Another crucial part of our study was to generate a DEB model for Bithynia tentaculata. DEB stands for Dynamic Energy Budget. This model looks at energy allocation patterns within an individual and where energy is allocated towards growth, reproduction, or maintenance. This model, along with statistical tests, serve as an important tool in determining differences between infected and non-infected individuals. In conclusion, through the collaboration of mathematics and biology, we are one step closer to understanding this host-parasite system as a whole and ultimately decreasing the amount of waterfowl deaths along the upper Mississippi River.